Today we're going to talk about some basic concepts of clinical microbiology. So we're going to talk about types of infectious organisms, sort of the spectrum of organisms that cause disease in human beings. We're going to talk about the process of infection, so the process in which an infection can move from one person to the next. We're going to talk about basic standard precautions and enhanced precautions for the control of infection, particularly as we see them in the hospital setting or in clinical settings. And we're going to also talk about the problem of antimicrobial resistance, where the drugs that we use to prevent infectious disease are actually becoming less and less effective because organisms are adapting to them. So we're going to talk about these issues, and I just wanted to explain a little bit about what you're looking at, this picture here, which I really am fond of. So what we're looking at is a petri dish, which is a plastic dish used in microbiology that is at the bottom of the dish we have this media which contains lots of nutrients and lots of food that microorganisms, particularly bacteria, can consume and grow. So a petri dish like this is where we culture a sample. So we put some sort of sample on a petri dish like this and we let it sit under controlled conditions and see what it grows. So what we have here, which is pretty obvious, is someone just put their hand on this petri dish and we just cultured whatever was on their hand. And this was, I assure you, what they thought was a clean hand. It may not be freshly washed, but it was a clean hand. And you can see that there's a wide spectrum of organisms growing, right? Lots of little colonies of different colored and different shaped organisms growing just from this sort of clean hand being placed on the culture media. And it really tells us a lot about microorganisms in our environment. We have a wide variety of microorganisms that are present on our body, on objects in our environment, and the majority of organisms are non-harmful to humans. The majority of microorganisms in our environment are non-harmful. There's actually only a small spectrum of microorganisms that are what we call pathogenic. And by pathogenic, I mean able to cause disease in human beings. And it's just this small spectrum of microorganisms that are actually the focus of clinical microbiology, meaning the application or understanding of these pathogenic microorganisms in the treatment of the diseases and the prevention of the diseases that they cause. So in this slide here, what we're looking at is a table where different classes of pathogenic microorganisms are laid out. Okay, so these are different classes, and then there's some descriptions of them. So we've got the relative size, so you can see that we're going from viruses, which are the very smallest infectious microorganisms, to helminths, which are a type of parasitic worm, which are actually quite large, right, up to 10 meters, right? They can grow very, very large. And then we've got the site of reproduction, so where these microorganisms live and reproduce themselves. And some of these microorganisms live inside of our cells, right? That's where they like to occupy, that's where they're going to reproduce. And the rest, most of them, occupy the extracellular environment. Okay? So they live in things like skin and mucous membranes and other locations. And then there are some examples of diseases that fit in these classes. Okay? So as you can see, there's a wide variety of classes of pathogenic microorganisms. Generally speaking, the classes that we see most commonly include viruses. Number one, they are the most common pathogenic organisms. Bacteria, right, which tend to be the second most common pathogenic microorganisms. And then to a lesser extent, but still more common than the rest, are fungi, right, fungal infections. So if we focus on those three for a moment and take a look at them, what we see here is some basic morphology or the physical structure of some of these microorganisms. So over here to the left, we've got different types of bacteria. So the bacteria that we are used to hearing in terms of disease-causing bacteria, the way bacteria are named are actually based on their physical structure. So bacteria are single-celled organisms. So they are single-cellular organisms that in their single-cell shape, they can have different basic shapes. So a spherical single-celled organism or single-celled bacteria we call a cocci. A rod-shaped or oblong-shaped single-celled bacteria we call the bacilli. And then this sort of almost worm-like shape we call a spirochete. Okay? Now, if you notice in the cocci, when these cocci grow in clusters, so when they divide and they grow in clusters, we actually call them, we call this form of reproduction, we call them a staphylococci. Okay? So these are spherical, single-shaped organisms that tend to grow in clusters. That is what a staphylococci is. Okay? And then in contrast to that, if you've ever heard the term a streptococci, these are spherical-shaped bacteria that tend to reproduce and form chains. Okay? So when you're looking at a sample, for example, cultured from someone who's ill, when they grow spherical-shaped cells that grow in clusters, we now know that this is a staphylococci, some form of staphylococci. And if they're forming in chains, we know this is some form of streptococci. Okay? So it's part of the identification of the bacteria. If we look over here to the top right, the B channel or B panel, we see a typical virus. Okay, so this is the shape of a typical virus. It's very, very small compared to the rest of the microorganisms. And viruses are non-cellular, so they are not composed of cells. In fact, a virus really challenges our idea of what a living organism is, because a virus is just genetic material. Okay, so at its core, it has some sort of genetic material. It can be a strand of DNA. It can be a strand of RNA. Okay, sometimes the RNA structure is double-stranded. Sometimes it's single-stranded. So it's got some sort of genetic material. And then surrounding the genetic material is a protein coat. Okay? And that protein coat can be quite elaborate, as you see in this picture, where there's both a protein coat and then a protein and glycoprotein capsule around it, okay, or envelope around it. But this virus, that's very simple in structure, must get inside of a host cell in order to reproduce itself. Okay, so viruses are really parasites in nature. They must infect a host cell and use that cell's protein manufacturing machinery, the cell's the contents of the cell, in order to reproduce itself. And now at the bottom here, what we see is a basic structure of a fungus, where fungi are multicellular organisms that they are sort of a cross between a plant and an animal cell. So they're very interesting in their reproduction pattern. But fungal infections tend to infect the surface of the skin or the surface of some epithelial structure. And they will form both spores, right? And these spores detach from the original fungus and travel through the air or travel by contact, and that's how they infect and spread. And they also, fungal infections or fungus in general in nature, also produce a quote-unquote fruit, which is what we observe as a mushroom. Okay? So in nature, when you see a mushroom, that is actually the fruit of some fungus. Okay. So viruses and bacteria are two important categories of microorganisms that cause disease in human beings. Now, with bacteria, when someone has a bacterial infection, we can go through some steps in order to identify the bacteria. The most important step is to culture the bacteria. So we take a sample, we take a sample, a throat sample or a sputum sample or whatever bodily secretion that we expect to see the bacteria, and we grow it in a petri dish. And then that way we can actually clearly identify what the bacteria is. Another part of bacterial identification is applying the Gram stain. And you may have done this at some point in some microbiology class that you've done previously. But the Gram stain is a very standard and very simple chemical reaction. It's sort of multiple steps. And it helps differentiate two major classes of bacteria, gram-positive bacteria and gram-negative bacteria. So very briefly, we've got our culture, we've got our sample of bacteria on this slide, and we're going to apply first a dye called crystal violet. Okay, so it's purple in color, and it's a dye called crystal violet. And as you can see, we've got a variety of bacteria here, some bacilli, some cocci, and they all get dyed purple in this first step. In the next step, we apply iodine. And what happens is the iodine attaches to the crystal violet and creates this very large structure for the dye. Okay? And that's a very important thing, because for some of these bacteria, that large structure gets trapped inside the cells, and for some other of the bacteria, it's still able to get out. Okay, so then we form this crystal violet iodine superstructure. The third step is that we apply this alcohol wash. And what the alcohol does is for some of the bacteria, it removes that color. Okay? For some of the bacteria, it's going to wear away on the first layer of the cell wall, and it's going to allow that crystal violet iodine to escape. Okay? And so you can see the bacilli in this slide have been decolorized, but the cocci remain violet. Right? So then the last stage is that we apply what we call a counter stain, which allows us to see the ones that were susceptible to the alcohol, okay? And it turns them pink. So now, after that final step with the application of the counter stain, what we clearly see are two colors that these bacteria take on. The original crystal violet, and then this saffronin pink. Okay? And the saffronin pink bacteria are what we call gram-negative, and the ones that remain crystal violet are gram-positive. So we now have identified two different kinds of bacteria. Now, what does gram-positive and gram-negative mean, and how is it significant in terms of clinical microbiology, in terms of disease? So if we take a closer look at these gram-negative versus gram-positive, okay, so the gram-negatives, remember, even though in this picture it looks purple, try to imagine it being pink, okay, because the gram-negatives were the ones that were susceptible to the alcohol and lost the crystal violet, okay. While the gram-positives were the ones that retained that crystal violet and remained purple at the end of the day. Now, if you notice, the gram-positives and the gram-negatives have very different cell membranes, structure, the outer structure of the cell. And that's exactly what we're looking at here. So both of these bacteria, if we look at the innermost part of their membrane, they have a classic plasma membrane, right, made of double layer of phospholipids and some proteins, okay? Now, the gram-positive, however, has this thick cell wall made of what we call a peptidoglycan, right? It's just a substance that forms this very thick cell wall in the gram-positives. Okay? The gram-negatives, they have a very thin layer of peptidoglycan, this very thin layer of substance making up the cell wall, and then they have an additional layer of membrane. Okay? Now, this difference, okay, is what creates this different susceptibility to the alcohol. When the alcohol is applied to these two membranes, in the gram-negative, the alcohol breaks down this outer membrane, okay, and what it does is it allows that crystal violet iodine dye to be let go. It allows that crystal violet iodine to escape. However, the alcohol can't penetrate this thick peptidoglycan wall in the gram-positive, so this cell retains the crystal violet iodine. Okay? So at the end of the stain, the gram-negatives appear pink, the gram-positives appear purple. Okay? Now, why do we care? Why is this important in terms of gram-negative and gram-positive? So most bacteria will differentiate between gram-negative and gram-positive. And in terms of treating human infectious diseases or understanding infectious diseases, gram-positive and gram-negative bacteria differ in some important ways. So if we look at the gram-positives, what we see is that there are far fewer pathogenic gram-positive bacteria. Okay? Most gram-positive bacteria are non-harmful, and there's really only a small group of gram-positive bacteria that are harmful. Okay? In addition, the gram-positives are more susceptible to an enzyme that we have in our body, particularly in saliva, called lysozyme. Lysozyme is an enzyme that breaks down that peptidoglycan wall and can destroy the gram-positive bacteria. Okay? So this lysozyme that we have in saliva, right, is effective or much more likely to kill gram-positive bacteria compared to gram-negatives. Additionally, most antibiotics will be able to attack and destroy gram-positive bacteria. So the gram-positives tend to be fewer of them are pathogenic. They are more susceptible to things like lysozyme and more susceptible to antibiotics. The gram-negatives, okay, in terms of clinical microbiology, they tend to be the bad guys. Gram-negatives, 90 to 95% of gram-negative bacteria are pathogenic. Okay? One of the reasons why gram-negatives are so disease-causing in humans is because in their outer membrane, they contain something called lipopolysaccharide. And you can see that right here, I'm pointing to it. Okay? It's this fat sugar complex that is attached to the outer membrane. It's lipopolysaccharide, LPS, also known as endotoxin. This substance, when it gets inside of our body, it activates a part of our immune system called the complement system. And the complement system triggers widespread inflammation, and it makes you feel very sick. Okay? So just to emphasize this, if I took LPS, endotoxin, without the organism at all, and I put it in your bloodstream, what you would see very quickly is the development of flu-like symptoms. Okay? So LPS, endotoxin, the outer shell of gram-negative bacteria, very strongly elicits a negative or toxic effect on our body. Okay? Fever, chills, aches, everything that you associate with being sick. Gram-negative bacteria are also far more resistant to lysozyme. Okay? Lysozyme, which breaks down that peptidoglycan layer. The gram-negatives, their peptidoglycan, they don't have a whole lot of them, and that lysozyme really can't cross that outer membrane. And the same is true with many antibiotics, that gram-negatives tend to have more resistance from antibiotics, and I'm not talking about developed resistance, just there are fewer antibiotics effective against gram-negative bacteria. And part of that reason is because antibiotics are very water-soluble substances, and they tend to not be able to cross this outer membrane. Okay? So those gram-negatives, in terms of clinical microbiology, they tend to be the bad guys more often than the gram-positives. So here we see that infections can affect many parts of our body, and based on location of the infection, you see here in this slide a list of more common microorganisms, pathogenic microorganisms that can cause infection in different sites of the body. Okay? Now this is purely for your reference, but one implication of this is, and it happens very commonly, is that people often will have an infection, let's say an inner ear infection, and they don't finish the penicillin or the amoxicillin that they're prescribed, right? So they have leftover antibiotic that treated their ear infection, and then later on maybe they get a urinary tract infection, and they think before they go to the doctor, or instead of going to the doctor, I'll just take this leftover antibiotic. And what's very important is that practice is extremely dangerous, because the microorganisms that tend to infect things like the head and the neck are very different from microorganisms that tend to infect the lower urinary tract or other parts of the body. Okay? So you'll find that drugs, different antibiotics used to treat different infections often differ greatly depending on where the infection is. Okay. So this process, this infectious process, it really describes the movement of an infection from one person to the next. Okay? And that's how infections spread. They're present in one person, and then it's transmitted to the next and transmitted to the next. Okay? Now, infectious diseases can be caused by a variety of microorganisms, as we said. Most common microorganism to cause disease are viruses. Okay? And the transmission of an infection is the requirements for an infection to be transmitted is often described as it being like a chain, where we have very specific steps that need to occur in order for an infection to move from one person to the next. So if we take a look at this analogy, the sort of chain of transmission of microorganisms, what we see is we start with a reservoir. Now, a reservoir is a organism or a place in which the microorganism prefers to live. Okay? Not where it can live, but where it prefers to live. Okay, so there are microorganisms that are very happy to live in humans. There are microorganisms that tend to live in animals, some in insects, and some even just in the soil. Okay, so an example of a microorganism that is very happy to live in an insect, because there's many, but of particular interest to us lately, is the Zika virus. right? So one example, the Zika virus. It likes to live in a very particular type of mosquito, right? Now, the next chain is how this organism is going to exit the reservoir. Now, it can exit carrying that Zika virus example. It exits through the oral mucosa of the mosquito. Okay? So the mosquito has the Zika virus in its saliva when it bites the person. Okay? So now we're on to mode of transmission. So for Zika virus, the mode of transmission is an insect bite, right? Is the mosquito bite, which transmits the virus from the saliva, from the oral mucosa to the person. Other modes of transmission include things like nasal droplets, when you sneeze and the pathogen escapes through droplets that are aerosolized in the skin. Another is semen. The HIV virus is found in high amounts in semen, and it can travel that way. So it then has to enter the next individual in some sort of port, right? So some port of entry. And that port of entry for our Zika example is a skin puncture from the mosquito. Okay, the skin puncture inserts that virus into the bloodstream. It can also be a port of entry. Can also be the nasal mucosa, right? Breathing in those droplets that somebody sneezed. The oral mucosa, those droplets getting in your mouth, or having a skin abrasion that allows the microorganism to directly enter. And then finally, one factor that affects this ultimate transmission is the susceptibility of the victim. So many microorganisms will be successful at transmission regardless of the susceptibility of the victim, but there are many factors that will enhance the likelihood of transmission. Things like having a victim that is malnourished, that is under-immunized, that is immune compromised. All of those things will greatly enhance the likelihood of transmission. Now, transmission of diseases, and we'll talk more about this when we talk about infection control precautions. Transmission is described in a couple of basic ways. There's what's called direct contact with body fluids, meaning you actually have to physically be in contact with it to be able to transmit the disease. There's transmission through droplets, meaning a person coughs or sneezes and the pathogen, that you have droplets that get aerosolized, right? And they contain pathogens, and then you can breathe that in and it can get transmitted that way. And then there's also airborne transmission, which is almost a extreme version of droplet. So with airborne transmission, those droplets that got aerosolized, the pathogen is able to actually stay in the air, persist in the air for much longer periods of time, and also is able to travel long distances. So basically breathing the same air that a person infected with this particular organism is breathing increases the chance of transmission. Okay? So in terms of these different modes of transmission, obviously airborne is an airborne transmission is the most worrisome because it is most easily transmitted. So in terms of infection control, trying to break the chain of transmission, we can break the chain of transmission by breaking any one of these steps. Okay? By disconnecting any one of these steps from the next step, we can actually reduce transmission of disease. So we can destroy the reservoir. Okay, so we can make sure that mosquitoes aren't breeding. We can make sure that we get good sewage treatment that reduces mosquito proliferation. It also reduces the likelihood of contamination. Okay, so we can destroy the reservoir. We can also block the port of entry. So we can use things like masks and gloves and isolation of individuals who are sick and condom use to prevent the entry of the organism. We can also block transmission by using sterile technique, by using hand washing, by cooking food. Okay? We can also block port of entry. Again, masks, gloving, condom use, and we can reduce susceptibility of the victim, right? Good nutrition, good rest, and vaccinations, extremely important in reducing the susceptibility of the victim. Okay, so all infectious control measures in the hospital, out of the hospital, are really based on this idea of breaking the chain of transmission at optimally all of these steps. Okay, so in a hospital setting, basic infection control procedures are described as infection control precautions. And as you can see in this visual, the precautions really start off with the basic precautions that we call universal or standard precautions, and then they get more and more enhanced. Okay, they get more and more enhanced depending on the particular disease and the pattern of transmission that the organism has for that disease. So standard precaution, but what's very important when in terms of understanding these precautions is that standard precautions are expected under all circumstances. So you notice that standard precautions is the first step. Okay? We can, no matter what else we're doing, we cannot ignore standard precautions. Everything builds on standard precautions. Okay? The next step is what we call contact precautions. And contact precautions are used when someone has a known disease that is transmissible through direct contact with bodily fluids. Okay? The next step is droplet precautions, where someone has a known disease that is transmissible through aerosolized droplets from things like sneezing and coughing. Okay? And then the most enhanced or the highest level of precaution is airborne precautions, where someone has a known disease that is transmissible through airborne transmission. Okay? So standard precautions prevail throughout, and then basically we're adding other layers to standard precautions depending on the microorganism and the pattern of transmission. So standard precautions. The idea is we are assuming that every person we come in contact with is potentially infected or colonized with an organism, and that it can be transmitted. So we just assume everyone has an infectious disease, and we take very standard precautions to prevent the spread. Okay? And there's two key components to standard precautions. Number one, most important, nothing else works unless you have good hand hygiene. Okay? So avoiding unnecessary touching of surfaces or unnecessary touching of patients, and performing good hand hygiene, meaning frequent washing of hands with soap and water, or at the very least with alcohol or hand sanitizer. Okay. So hand hygiene is the first and foremost step for all infection control. Additionally, we have the use of what we call PPE, or personal protection equipment, and they include things like gloves, gowns, mouth, nose, and eye protection. So under standard precautions, we are using gloves if we're going to come in contact, our hands are going to come in contact with blood or any bodily fluids. Okay, whether we're assuming everyone has the disease that's transmissible, so if we're going to come in contact with blood or bodily fluids, we're going to use gloves always. Okay, we're going to use gowns if we're performing some procedure where we anticipate the contact with blood and bodily fluids. Okay, where blood and bodily fluids can actually be sprayed and enter and get on our clothing or something like that. So we're going to wear gowns depending on that. And then additional PPE includes covering the mouth, the nose, or eye protection if we think that the procedure we're performing on somebody can cause the splashing of blood and bodily fluids onto our mouth or our eyes or our head. Okay, so the use of personal protection equipment is setting specific and activity specific. But under standard precautions, if we're going to come in contact at any of these levels with blood or bodily fluids, we're going to use this equipment to protect ourselves. Contact precaution, okay, in addition to standard precaution, is used when we have a patient. Now doesn't have to be assumed. We know that a patient has an infectious disease that is transmissible by direct contact with bodily fluids. Okay, so a person, for example, has a wound on their leg that is infected with methicillin-resistant staphylococcus, MRSA. Okay, now that patient is going to be put on what's called contact precautions, and that means that they're likely going to be put in a private room so that they can't infect their roommate. Anyone entering the room is going to have to wear gloves and gowns. Okay, anyone entering the room is going to wear gloves and gowns. Anyone entering the room, right, this is part of standard precaution, is going to wash their hands immediately before and after they put on their gloves and gowns. We're going to limit transport of this patient outside of the room so that we limit opportunity for spread. Also important, any equipment that we use on this patient, we're going to prefer to use disposable things. Or we're going to use things that are left in the room. So for example, your stethoscope, many nurses and doctors, they carry their stethoscope that they prefer to use around their neck, and they use it from patient to patient. And of course they are cleaning the stethoscope between patients, or at least they should. With contact precautions, there's going to be a stethoscope in the room that never leaves that is going to be used on this patient. Okay, to prevent the spread of this disease from patient to patient through the movement of the stethoscope out of the room. And then environmental measures, this room is going to be prioritized for frequent cleaning and desanitation. Okay? That's contact precaution. One step up from contact precaution, and it piggybacks on contact precaution, is droplet precaution. And this is when a patient has a disease where blood or bodily fluids can get aerosolized in the air, right, into these droplets in the air through things like sneezing or coughing. Okay? And so with droplet precaution, in addition to standard precaution, and really in addition to contact precaution, we're also going to have people wear a mask, okay, to prevent the droplet from being inhaled or ingested into the person's nose or mouth. Okay, so droplet precaution on top of standard and contact, the person, anybody, everyone who enters the room, in addition to gowns and gloves, are also going to wear a mask. Okay. airborne precaution, this is where those aerosolized droplets, okay, the particular disease now is so easily transmitted that the droplets can be so tiny, and the pathogens suspended in those droplets can persist in the air. They can linger in the air for a long period of time, and they can travel a long distance. So basically breathing the same air as this patient is leaving anyone highly susceptible to acquiring the disease. So with airborne precautions, it's the highest level of precaution. It's building on standard, contact, and droplet. The patient is placed in a private room, and the door is always closed. And this room is a special room. It's a room with negative air pressure, which means that the air in the room is actually constantly getting sucked out. And basically, between 6 to 12 times an hour, the air that occupies this room is going to be completely refreshed. So, and that we have HEPA filters that are used at the suction point and the reentry point for air circulation. Okay, so this patient private room, every hospital has at least some of these negative air pressure rooms, they're basically maintaining a very carefully controlled air quality. In addition to that, right, with airborne precaution, everyone who comes in the room is also going to wear a very special kind of mask called an N95 respirator mask. Okay. Now this N95 respirator mask is actually something that needs to be fitted from person to person. So you are fitted to make sure that there's no opportunity for air to get around it. It specifically fits your face. Okay. So in addition to the negative air pressure room, you get this very individualized, customized respirator mask. And the movement of the patient is highly limited. Okay? Everybody comes to the patient. So those are the levels of precaution that you will observe in a hospital setting. Now, when we treat infectious diseases, we use a whole host of drugs that are called antimicrobial drugs. And this includes drugs that are used to fight viral infections, drugs that are used to fight bacterial infections, fungal infections, and the entire spectrum of microbial drugs. Now, there's an interesting thing that happens with the use of antimicrobial drugs, and that is whenever we use a drug like this, what we're actually doing is creating evolutionary pressures for the microorganisms to actually adapt and develop a resistance to the drugs. Okay? And this is something that happens rather quickly because bacteria and viruses, for example, they can mutate and evolve very, very quickly. Their evolutionary time is on the order of days to weeks. It's not, centuries to millennia like humans. They evolve very, very quickly. So what we've found, and it's a growing problem worldwide, is how common the appearance of antimicrobial resistance is. And this is true for resistant viruses and resistant bacteria. But what we tend to talk about a little bit more is the development of antibiotic-resistant bacterial infections. So in this slide here, what we're looking at, just to really drive home the idea that this happens really all the time and it happens quickly, is if we look at the history of the treatment of Staphylococcus aureus, a very, very common bacterial infection The very first antibiotic ever developed, penicillin, 1941. Within a year of the development or introduction of penicillin, we first started to see penicillin-resistant Staphylococcus aureus. Okay, and then the different generation of drugs that were developed subsequent to penicillin to fight Staphylococcus aureus, streptomycin, tetracycline, erythromycin, gentamicin, methicillin, right? They were all introduced at later dates from 1941. And within a couple of years of their introduction, we first started to see resistance to those drugs. So resistance develops quickly, and it develops nearly universally, okay? And that really presents a problem. But what we definitely know is that the more we use an antibiotic, so the more overuse and inappropriate use of an antibiotic there is, the faster and more widespread the development of antibiotic resistance there is. So if we project by the year 2050, what we see are astounding numbers of deaths projected deaths attributed to antimicrobial resistance worldwide. Okay, so this is really a worldwide problem. And if we look at, from the perspective of a pathogenic bacteria, what we see is that what they develop in order to become resistant to antibiotics is a variety of strategies to basically evade these antibiotics. So these are just some examples. So here we've got our bacteria, and some examples of how the bacteria managed to become resistant is in, for example, decreasing the influx or the uptake of the antibiotic. So sometimes antibiotics require a binding to a receptor on the cell in order for the drug to be taken up. So some bacteria just reduce their expression of those receptors, so the bacteria doesn't enter. Another very common mechanism for resistance is inactivating the antibiotic. So the antibiotic enters, but the bacteria has developed an enzyme that actually breaks it down before it can attack the bacteria. Okay? So the bacteria develops, there's a gene that mutates, and it develops this gene that can code for an enzyme that breaks down the bacteria. Okay? Sometimes bacteria will just, as the antibiotic enters, it will just push the antibiotic out. Right? It's a process called efflux. Okay? Sometimes the target that the antibiotic has inside the cell, the way it fights the bacteria just modifies the target so the antibiotic can't recognize it, or it just boosts that function so the antibiotic actually can't overcome that amplification. Okay, so lots of different ways or lots of different mechanisms that we've identified for bacteria to be able to resist antibiotics. And if you notice, these are in the white boxes, they're example antibiotics for these different mechanisms. And if you notice, there's one name that comes up over and over again, beta-lactams. Okay, so beta-lactams over here, beta-lactams here, here. The beta-lactams include the family of penicillins. So the beta-lactams are the oldest category of antibiotics, and as you can see, bacteria have developed the greatest number of mechanisms to evade beta-lactams, which doesn't mean that they're not still effective. It just means that it just sort of drives home this idea that the more experience bacteria have with an antibiotic, the more it will develop, more likely that resistance will develop. Now, this idea that the more overuse or inappropriate use of antibiotics, the more likely we see resistance. We normally actually think about this in terms of clinical practice. We think about it in terms of medicine, that when you are sick and you go to the doctor and you insist on getting antibiotics, even though you have a viral infection and the antibiotics can't fight that viral infection. So that does increase the likelihood of resistance. However, if we really are honest and we look at the annual estimated antibiotic use in just our country, okay, what we find, the red piece of this pie is all antibiotic use in humans. And then the blue piece, which is the vast majority of this pie, is antibiotic use in livestock. And in fact, the vast majority of antibiotic use is in agricultural settings. So, and it's not even always to treat ill animals. There's actually many, many examples of the use of antibiotics on animals that are not ill, right? They're used for prophylactic reasons, to prevent infection, to enhance the likelihood that animals will be able to be transported in a healthy way. So these antibiotics are used widely in agriculture with livestock, and there is a growing concern that the rise of antibiotic resistance is really required. In order to control that, we have to address this use of antibiotics, not just in clinical settings, but also in terms of agriculture applied to livestock.